Hi friends, today I'm welcoming you all to the my new channel, that is Learn About Medico channel. Today I'm going to introduce about the vitamins. About vitamins, we are going to introduce about the vitamins. The vitamins, the vitamins are nothing but these are the chemical substances. The vitamins are the chemical substances which are help in the our biological functions, and these are obtained from the dietary sources. These are nothing but vitamins. The vitamins are nothing but the biochemical substances which are required for our biological functions, perform the normal functions of our different functions of our body. Okay, the vitamins are mainly classified into two types. They are going to study under the two headings. These are, first one is fat soluble vitamins, fat soluble vitamins, and another one is water soluble vitamins. Another one is water soluble vitamins. Okay, in the fat soluble vitamins, we are having vitamin A and vitamin D and vitamin E and vitamin K. Under the fat soluble vitamins, the fat soluble vitamins are vitamin A, vitamin D, vitamin E, and vitamin K are the vitamins which are fat soluble. And then come to the water soluble vitamins. In the water soluble vitamins, we are going to study about under the two headings, that is non-B complex vitamins, non-B complex vitamins and B complex vitamins. Non-B complex vitamins and B complex vitamins. Water soluble vitamins, non-B complex vitamins as well as B complex vitamins. In the non-B complex vitamins, the example is vitamin C is the non-B complex vitamin. In B complex vitamins, we are having vitamin B1, and vitamin B2 and vitamin B3 and so on, and vitamin B12 as well as folic acid, and vitamin B1 is also called as thiamine, and vitamin B2 it is also called as riboflavin, and vitamin B3 is also called as niacin, and so as the vitamin B12. These are the classifications of vitamins. In that, today we are going to discuss about mainly about the vitamin A. The one by one we are going to discuss. In that, first one is vitamin A. We are going to discuss briefly about separately the vitamins. That is, today we are discuss about vitamin A. Okay. The vitamin A, it is in plants. The vitamin A in plants. This is in the form of beta carotene. And in animals, the vitamin A in the form of retinol and retinoic acid. Okay, the vitamin A which is present in the plants in the form of beta carotene. In the plants it is in the form of beta carotene, and in animals it is in the form of retinol, and retinoic acid. This is all about introduction about the vitamin A. For every vitamin, we are going to study the first about the chemistry part, the chemistry of the vitamin. First, we are going to study about the chemistry of the vitamin, and after that, RDA, that is recommended dietary allowance, and after that, we are going to study about the dietary source and dietary source, and after that we are going to study about biochemical functions, the biochemical functions, and after that the deficiency manifestations. After that the deficiency manifestation. The deficiency manifestation. For every vitamin, we are going to study the stepwise. Like first, we have to study the chemistry of the vitamin, and the RDA of the vitamin, and dietary source, and the biochemical functions, and deficiency of the vitamins causes which are the diseases. First, we have to discuss about the chemistry part of the vitamin A. We have to discuss about the chemistry part of the vitamin A. Previously, I have told you that is what, that the vitamin A in animals, it is in the form of retinal, NAL. It is in the form of retinal and retinol. Second one is retinol, and third one is retinoic acid. In animals, that the vitamin A, that is in the form of retinol, and retinoic acid. This retinol, it is the aldehydic form. That is aldehyde form. This is the aldehyde form of vitamin A, and retinol, this is the alcoholic form. This is the alcohol form. And this retinoic acid, this is the carboxylic acidic form. That is carboxylic acid. That is the retinol, this is the aldehydic form of vitamin A, and retinol, this is the alcoholic form of vitamin A, and this retinoic acid, this is the carboxylic acidic form of vitamin A. Okay, after this, we move on to the retinal, if it undergoes oxidation, the retinal undergoes oxidation, which gives retinoic acid. How we are going to obtain that retinoic acid? When the retinal, it undergoes oxidation, that gives retinoic acid. I will show you with the chemical structure. Okay, I will show you with the chemical structure. CS3, double bond. Here is CS3, that is methyl group. Here is also CS3. Double bond, and here E double bond OH. This is the structure of retinal. That is, this is the structure of retinal. I told you people, that is, retinal undergoes oxidation, which gives retinoic acid. This is the retinal structure. It undergoes oxidation, which gives retinoic acid, Okay, oxidation means, in general, it is the addition of oxygen. Oxidation means the addition of oxygen or removal of hydrogen. Here, this oxygen is going to attach to this carbon atom. That is, the carbon is there. That is C double bond O. That to this carbon atom, this oxygen is going to be added, and it becomes OH. This oxygen is going to be added to this carbon atom. It becomes OH. And here, I will going to write the same structure again. Here CS3. Here is also CS3. Double bond. Here is CS3, and here is also CS3. What happened here? Oxidation means addition of oxygen. This carbon atom is added with this oxygen. That is C double bond OH. C double bond O is as it is. This oxygen and the hydrogen combined to form OH. This is the structure of retinoic acid. That is, the retinal undergoes oxidation, which gives retinoic acid. And after that, second one is, if this retinal, it undergoes reduction. If the retinal undergoes reduction, it gives retinol, Okay, retinal undergoes reduction, it gives retinol. I will show you with the structure. Okay, as you know, this is the structure of CH3 retinal. Double bond, double bond, double bond, double bond. Here is CH3, here is also CH3, and here C double bond O. This is H. This is the structure of retinal. This is the structure of retinal. If this retinal undergoes reduction, reduction means in general addition of hydrogen, or removal of oxygen. If this retinal undergoes reduction, it gives retinol. What happens, you know? Here, this double bond is going to be cleavage. It becomes single bond, and this H2 is going to be added to this carbon atom. This H2 attached to this carbon atom becomes CH2, and this double bond cleavage, that is breakdown. This O and H combines OH, that is CH2OH. I will write here. You can see here. CH3, double bond, double bond, double bond, double bond. Here is CH3, and here is CH3, and this double bond is cleavage, and this H2 is added to this carbon atom. This OH, O and H is combined to form OH, that is CH2OH. This C and CH2 combines, and O and H combines CH2OH. This is the structure of retinol. That is, retinal undergoes oxidation, it gives retinoic acid. If that the retinal, it undergoes reduction, it gives retinol. This is all about what? Retinol, and retinoic acid. Then I told you people that the vitamin A in plants, that the vitamin A, it is in the form of beta carotene. Beta carotene, it is nothing but it is the combined form of 2 moles of retinol. That is retinal plus retinal. Retinal plus retinal is nothing but is beta carotene. 2 moles of retinal, 1 mole and then 1 mole. 2 moles of retinal combined together, that is beta carotene. We can obtain this beta carotene in the form of plant foods. In the form of plant foods, we are going to obtain, and in the intestine, that is the beta carotene. In the intestine, that is the beta carotene. Beta carotene means the 2 moles of retinal combined together form beta carotene. In intestine, this is beta carotene is going to be cleavage. That beta carotene in intestine, it is going to be cleavage. The beta carotene cleavage, that the 2 moles of retinol combined together form beta carotene. In intestine, this beta carotene going to be cleavage to form two moles, two different moles of retinal separately, and another 1 mole of retinal. That is beta carotene, it undergoes cleavage to form two moles of, that is retinal and retinal. To form these retinal, that is the beta carotene, it undergoes cleavage. This is all about the chemistry part of the vitamin A. RDA. That is nothing but recombinant dietary allowance. RDA. The RDA of that vitamin. RDA is nothing but the recombinant dietary allowance. The recombinant dietary allowance means it is the quantity of the nutrients. The quantity of the nutrients which are required through the diet, which are required daily through the diet, these are called as the recombinant dietary allowance. For the vitamin A, it is in adults, the vitamin A RDA is 600 micrograms per day, and it increases in the increases in pregnancy and lactating mother. Okay, the RDA in adults it requires the 600 microgram per day and it increases during the pregnancy as well as lactating mother. This is all about the RDA of that vitamin. After this, we know about the sources of the vitamin A. The sources of vitamin A. In that sources, we are going to study about literally in the plant sources as well as the animal sources. In plant sources, the yellow and dark vegetables. The yellow and dark vegetables. The yellow and dark vegetables are the good plant sources of vitamin A. For example, carrot, pumpkin, mango, and papaya, etc. The plant sources, yellow and dark vegetables are the good sources of vitamin A. For example, carrot, pumpkin, mango, and papaya are the good examples for the plant sources. And in the animal sources, the liver, kidney, and milk, butter, and liver oil. Liver oil is the good source. The liver oil is the good source of vitamin A. This is all about the sources of vitamin A. After this, we move on to the biochemical function. The biochemical functions of vitamin A. Biochemical function. The biochemical function. These functions we are going to study under the heading of two headings. That is, first we have to know about the retina of the eye. Retina and Wall's visual cycle. In this function, we are going to discuss about the retina as well as Wall's visual cycle. Or this is also Wall's visual cycle, also called as rhodopsin cycle. Okay. First, we are discuss about the retina of the eye. This is the structure of eye. Just assume if it is the retina of the eye. This is what? The retina. The retina of the eye. Retina of the eye. In the peripheral part of this retina, it is contains the kinds of cells. In the peripheral part, it is going to contain a kinds of cells. These are called as rods. In the periphery of the retina, these contains a kinds of cells called rods. And in the center part, these are contains the conical shape, the cones-like structures. These cells are called as cones. Okay, the retina contains two kinds of cells. The retina contains two kinds of cells. First one is rods, the peripheral rods and inner central cones. The rods are peripheral cells. Peripheral cells, and cones are central cells. In the human being, the rods are nearly 10 millions of rods are present. 10 million rods. In the human being, nearly the 10 millions of rods are present in our retina, and nearly 5 million nearly 5 million of cones are present in our retina. Okay, the function. What is the function? We are going to discuss in above. Rods and cones. These are 10 million. These are 5 million in human being. And the rods which are responsible, and these are involved in the dim. These are involved in the dim light vision. The rods are responsible for the dim light vision. Okay, and cones which are 5 million in the human beings, and these are responsible for, and the cones which are involved in the bright light vision, and these cones are involved in the bright light vision as well as color vision, as well as color vision. Okay, the rods which are involved in the dim light vision and cones which are involved in the bright light vision as well as color vision. For the color vision, these cones are having the pigments. For the color vision, the cones are having the pigments. These are having the pigments. That is, the first one is porphyropsin, the pigments of the cones. For the color vision, the cones are having the pigments. That is, first one is porphyropsin. This is responsible for the red color vision. The red color vision which is done by this porphyropsin. And second one is iodopsin. Which is responsible for the green color vision. The iodopsin which is responsible for the green color vision. And third one is synopsin. The third one is nothing but the synopsin which is responsible for the blue color vision. Okay, the pigments, the cones are having the pigments for the identification of the color. That is, the pigments, first one is porphyropsin which is responsible for the red color vision, and iodopsin which is responsible for the green color vision, and synopsin which is responsible for the blue color vision. After this, we move on to the Wall's visual cycle. The Wall's visual cycle. The name, the Wall's is nothing but, this is the Jazz Wall's. The Jazz Wall's is nothing but it is the name of the scientist. That's why he invented this cycle. That's why the name is given Wall's visual cycle. And this is also called as rhodopsin cycle. Okay, the rhodopsin is nothing but, okay, I will write separately. Rhodopsin. This rhodopsin is nothing but, this is the conjugated protein. The rhodopsin is nothing but, this is the conjugated protein with the combined form of 11 cis retinal plus opsin protein. This is the protein. The rhodopsin which is the conjugated protein with the combined forms of the 11 cis retinal and opsin. Okay, the cycle, that is the rhodopsin cycle, it starts from the rhodopsin. Okay, that's why it is named as rhodopsin cycle. Okay, this rhodopsin, it is going to give, once the light enters into our retina, once the light, that is the photon which enters into our retina, this mechanism, this cycle is going to be start. This rhodopsin, once it receives the light from the retina, it gets dissociated with, it is going to be released opsin protein, and this rhodopsin is converted into all trans retinal, all trans retinal, NaL, all trans retinal. The rhodopsin converted into all trans retinal. Once the light is enters into the retina, this rhodopsin is converted into all trans retinal by releasing of this opsin. Okay, this immediately the all trans retinal, NaL, retinal is going to be converted into 11 cis retinal, 11 cis NaL, retinal. The all trans retinal, it is going to be converted into 11 cis retinal with the help of enzyme retinal isomerase. The all trans retinal is converted into the 11 cis retinal with the help of enzyme retinal isomerase. And this 11 cis retinal, it is going to be combined with the opsin. 11 cis retinal, again it is going to be combined with the opsin and gives rhodopsin. Rhodopsin is nothing but it is the combined form of 11 cis retinal and opsin. The rhodopsin is undergoes, once the light is entered into the our retina, it is get converted into all trans retinal by releasing of opsin, and this immediately the all trans retinal is going to be converted into 11 cis retinal with the help of enzyme retinal isomerase. This 11 cis retinal, it is going to again combined with the opsin and gives rhodopsin, or 11 cis retinol plus opsin, which is the combined form called the rhodopsin. And it is the mechanism of the vision. It is not the complete mechanism of vision, and also in the liver it is incompleted. To complete that vision mechanism, this all trans retinal, to complete that vision mechanism, this all trans retinal is transported into the liver. It is transported into the liver. In the liver, this all trans retinal is converted into all trans retinol, NOL. This is retinal, NaL. This is the aldehyde form of the vitamin A, and this is the retinol, OL. Retinol, it is the alcoholic form of the vitamin A. In the liver, all trans retinal is converted into all trans retinol with the help of enzyme alcohol dehydrogenase. By utilizing of NADH plus H plus, it acts as a coenzyme. This NADH plus H plus, which converted into NAD plus. In the liver, this all trans retinol converted into all trans retinol with the help of enzyme alcohol dehydrogenase. By utilizing of this NADH plus H plus, it is going to be converted into NAD plus. Okay, and again this all trans retinol converted into 11 cis retinol. Okay, all trans retinol converted into 11 cis retinol with the help of enzyme isomerase enzyme. This is also happens in the liver. This is also occurs in the liver. The all trans retinol converted into all trans retinol. This is also in the liver, and all trans retinol is converted into 11 cis retinol with the help of enzyme isomerase. This is also occurs in the liver. And again, this 11 cis retinol is converted into 11 cis retinol. Retinol, OL, is converted into AL. 11 cis retinol is converted into 11 cis retinal with the help of enzyme, same enzyme, alcohol dehydrogenase. Here the NADH2, which is utilized, here it is going to be again produced. This is NAD plus is converted into NADH plus H plus. This is all about the Wolff visual cycle. The rhodopsin converted into all-trans retinol once the light is enters into our retina, and by releasing of opsin, this is immediately converted into 11-cis retinal with the help of enzyme retinal isomerase, and again this 11-cis retinal combines with opsin and gives 11-cis rhodopsin. And this is not a complete cycle. That's why to complete that vision mechanism, that all trans retinol is converted into all trans retinol with the help of enzyme alcohol dehydrogenase, and that all trans retinol is converted into 11-cis retinol with the help of enzyme isomerase, and this 11-cis retinol is converted into 11-cis retinol with the help of enzyme alcohol dehydrogenase. This is all about Wolff visual cycle. After this completes the biochemical functions of vitamin A. After the discussing about the functions, we move on to the deficiency manifestation. Okay, now discuss about the deficiency manifestation of vitamin A. In that, first one is night blindness. Night blindness. It is the condition in which the night blindness is nothing but it is difficult to see. It is difficult to see dim light. Next one is xerophthalmia. Xerophthalmia, it is nothing but the retina becomes opaque. Retina becomes opaque and rigid. The retina of the eye which becomes opaque and rigid, and it becomes dry. The condition, the retina becomes opaque, rigid and the dry, this condition is called as xerophthalmia. And third one is Bitot spot. This is the condition where the retina and the conjunctiva cells, in that the triangular like structure, the plateaus, the triangular like plateaus which are occurs in that cells, that condition is called as Bitot spot. And next one is keratomalacia. This is occurs due to the deficiency of keratin. And accumulation of keratin of keratin, which leads to the keratomalacia. It is the condition where the deficiency of keratin is occurs, and the accumulation, the deposition of keratin in the retina. This condition is called as keratomalacia. This is all about the vitamin A. Thank you.